this session from around the country. Uh, this is extremely special because this is our first scientific CME, and this is quite a unique CME also because of the topic and our faculty both as well. So this is probably first of its kind such event happening in India, and we are very lucky for Maharashtra Ophthalmological Society to have it under aegis of MOS. Global Eye Genetic Consortium around the world is the organization which looks after the eye genetics, its implications, its research. And here in Maharashtra Ophthalmological Society, we are honored that they decided to collaborate with us for this hybrid CME, and which is going to be followed next weekend by the first international conference of the Global Eye Genetics Consortium, which is a great honor for MOS. And this meeting is going to happen in Bombay Hospital in Mumbai. And please also consider this to be our warm invitation for all of you to please attend this meeting as well. So today is the meeting, which is a CME. This is happening in Ruby Hospital under the aegis of MOS and POS in association with Kamala Sundaram Foundation in Mumbai, which is the flag bearer is our favorite Dr. Nati sir, and Ruby Hall Clinic in Pune. We are going to deal with the genetics and the eye, and the whole purpose of conceptualizing this meeting was to have all our clinician ophthalmologists to have certain basic knowledge why the knowledge of genetics is important, and in their clinical practice, can it influence the outcome of our patients? If there is any patient who has genetic disorder, can we guide them better? Can we give them much better counseling, much better treatment because of our knowledge of today's CME? And we are extremely honored to have with us today Dr. John Gyan Prakash. He is the director of the Office of Internal Program Activities of the National Institute of Health in USA, Washington. And Dr. S. Natarajan sir, who is the Secretary General of the Global Eye Genetic Consortium and the Chief MD of the Aditya Jyot Eye Hospital, and of course, the past president of All India Ophthalmological Society and Maharashtra Ophthalmological Society. We also have Dr. Kumudi Godbole ma'am. She's the consultant clinical genetist with us. And of course, we have with us our president, Dr. Santosh Bide sir. So I would like to request Dr. Bide sir to kindly felicitate our faculty, our international faculty, Dr. John Gyan Prakash sir. He has come all the way from Washington for this meeting for today, and also next weekend, the first international consortium that we are going to have. So Dr. Bide sir, can you please kindly felicitate Dr. John Gyan Prakash sir. Please give a round of applause, and thank you sir for taking time out for MOS and POS. I would like to request Dr. Bide sir to now felicitate our favorite Dr. Nati sir, who is ever energetic and inspiring figure for all of us, and especially for me since very young age. So please give him a round of applause. It is actually Dr. Natarajan sir's efforts that we are going to have today's CME and also the conference next weekend. Can I request Dr. Bide sir to also felicitate Dr. Kumudi Godbole ma'am, who has taken her time out, and she is going to share pearls with us as a consultant clinical genetist. Thank you very much, and I would like to invite all the guest faculty and the office bearers for the lamp lighting ceremony for 2 minutes, and after that we are going to start the CME immediately. Thank you.
We can give it to somebody, into somebody from. Past president of POS.
Yeah. I'm the only POS representative.
Yeah, Santosh is there. I'm also somewhere, I don't know. Yes, sir. Yeah, one, Poona. So Vardhaman will do next. Yes. Even and you want to come, madam. Come, yeah. And then Jeevan. Yes, everything is well planned. And Jonas.
We will wait for some time. We are loading with the presentation and then we'll start. Which one? This one? Who's the first person? Dr. Prakash. Is the copy. So good evening, everybody. So let's start with our first lecture by our guest speaker from USA, Dr. John Gyan Prakash. He has done his BSc from University of Allahabad and MSc in biochemistry from University of Allahabad. After that, he has done his PhD from University of Illinois Urbana-Champaign. Currently, he's leading the international program and global health initiative at the Office of Director of National Institute of Health, that is NIH. He has also received many awards. He has traveled all over world. He has received fellowship of UNESCO. He is the founding president of Global Eye Genetics Consortium, and as Dr. Vardhaman has already said, that under aegis of MOS, we are going to have our first international conference of GEGC in Mumbai next weekend, coming weekend, that is on Saturday and Sunday. So Dr. Gyan Prakash is going to talk on opportunities for research and training in eye research and participation of clinicians in research programs. So Dr. Gyan Prakash, please.
So good evening. It's such. Yeah, let's put it full screen. Should be here. It's full screen. Niche, na?
Yeah, I'm just
Remove this up.
one.
Yes, it. Yeah,
One.
Okay. I'm gonna leave this.
Open the screen. Have you shared this?
Yeah, it's on sharing. It's on sharing.
Now you are spared.
If we go to play, full screen here. This one.
Yeah.
This bar is over here. That's the problem.
We'll see. Yeah.
Black thing, no? That stops and you can
No,
move.
no, F5.
Okay, one more.
No, no,
Down, no.
It's
You can move this up.
Now you go to slide
I move it up.
1 minute.
from
Ah,
current.
correct.
slide.
Now you
correct,
can see
correct,
it.
correct.
Move it totally up.
Right, right.
No, excuse me.
Yes, sir. 1 minute. I'll just move it up. 1 minute. One.
Oh,
Thank you.
But in everything, sir.
Yeah, thank you so much, Dr. Natarajan, for your technical assistance. You are always available for all the technical assistance. But clearly, let me start out by just saying that it's an honor and a privilege to be in Pune. This is my first day actually in Pune. So I literally arrived, Dr. Natarajan and I arrived here from Delhi past midnight, right? So this is our first day. We can technically call it our first day. And clearly it's, I've been discussing with Dr. Santosh, Dr. Vardhan, and, the whole team. So I'm here now. So and my talks are generally very informal. So, and you'll just next few minutes, you'll see that, I really want to, give you a message, more of a strategic vision. And then Dr. Natarajan and Dr. Kumudi will get into more, some of the details of how you can actually practice and bring genetics into the local and your clinical practice. So I'm going to start by thanking the Maharashtra Ophthalmology Society, MOS first. And then, of course, Professor Natarajan, who is, he tells me to come to India, and wherever he takes me, I'm here. So I report to him. As soon as I land in India, I have to make the first phone call to Dr. Natarajan. So I'm here. What do I do, sir? So he, so I'm just following in his footsteps. But amazing, he's a family, he's a friend. And then now I have new family members. I'm related to Marathis today, right? Of course, then I showed you the pictures, right? So what I do is I'm the director for the National Eye Institute in an international office. And the most importantly, I'm going to show you that what the big picture is, because you really, in America, we say this is where the action is, where rubber meets the road, right? And I'm sure the people who have been in America, they know that. But let me take you on a bigger journey, bigger perspective, and a strategic perspective. And then we can drill it down. So by the time the evening finishes, you will have a clear picture of what is going on at the worldwide level or the country level and so on. So let me start my journey here. Next about 15 minutes or so. Okay, so no financial disclosure. And the most important thing, since I do informal presentations, a lot of this is my own, and the government doesn't take responsibility. So I have to put this on. We know that. I'm not going to go into details. We know the global blindness is a serious problem, big problem. 2.2 billion, definitely is more than that. And clearly, the take-home message here is that the bigger portion bigger burden is in this part of the world than where I am right now. So we need to bring a larger collaboration with clinicians, with researchers, and everybody else who can work with this. And as a scientist and clinicians and researchers, we don't know any boundaries because we work with the people, their disease, and so on. And these boundaries, in our mind, really don't exist. Because if you find something, a new finding, it helps us as well. And if you find something, it will help you. So, and an important thing that India and US are not only two largest democracies, but also two largest eye research communities. The bigger, if you go to any international meeting, who are the two countries and and participants, researchers, medical experts? Number one, US and India, of course, large number. So we have a lot of potential. So give you a, and I'm not going to go through all this in detail, But I just want you to be aware, we have new leadership. Dr. Michael Chang is the new director, and Dr. Santa Tuminia is the deputy director for National Eye Institute. Important thing is that we have just released our new strategic plan, and I put the scan. You can scan this and look into the details. But we literally have put in all the traditional areas of eye diseases. But important thing is that we have put these cross-cutting areas, and that's where the growth will be. From genes to disease, from biology to neuroscience, immune systems, regenerative medicine, data sciences, artificial intelligence is just becoming this going to be a very important tool, quality of life, and then disparities research, unique populations, and so on. So I would like you to take a look at someday, just go on this website, National Eye Institute, and take a look at this. This will give you a worldwide perspective, and I urge you to do that. Now, I want to give you a quick run of what we do at National Eye Institute. Basically, this was developed on a piece of paper napkin and became a slide. So there are essentially 3 different areas. Largest part is a research program, then training programs, and then special projects. Let me start from bottom. A special project, such as, our institution played really key role in developing COVID vaccine, right? National Institute of Health. You may have heard of my previous boss, Dr. Anthony Fauci, right? And his office is right above my office, actually. So clearly, and I'm used to seeing him pretty much every day. So that's sort of mandate. NIH is a very unique organization. The overall budget of NIH is about $52 billion a year. Think about it. So there are 27 institutes within NIH. One of them is National Eye Institute, somewhere budget-wise in the middle of one of the 27, right? I'm just giving you a big picture. Our budget is about $865 million, just for what? For eye research, right? So very unique. Whatever we do, it sets the standard and direction for anything what people do all over the world for eye research, right? So that's why the strategic plan is very important. Huge, big perspective. So this will give you an idea. Our NIH director recently said that we have 14,000 collaborations around the world. Now, what do we do this is NIH wide, right? So out of the 27 institutes. Now for any eye, if you look at that, so when I started about this is about 5-year-old data, but clearly things were just beginning in terms of international collaboration. Now we have relationship in 55 countries, and India is one of the 55 countries. Over about 600 collaborations, and we are spending clearly a lot of money researching, working with collaborators all over the world. This actually gives you countries where the top collaborations are taking place. So, but there's a huge area where still there's no collaboration. And one of my goals is to really bring the unique patient population. This is, again, it gives you where India is right now. It's definitely one of the top 10, but we are working to really make it a even bigger program. So these are some of the examples of research collaborations. As you can see that Indo-US program, that's what I'm going to focus. The India government and the US government, we signed a deal where we actually work with the government of India, and we bring the Indian and US investigators together, and we fund new programs in pretty much all areas of research. Now, let's move on to the genetics. So about 40%, 38% of our current grants, current programs relate to somehow, somewhere related to genetics. So obviously, this is really not only has come up, but in the research circle, this is really moving very fast. 4 out of 10 programs are moving into genetics program. Now, when people do research, what do they look for? Impact factors. Some of you are researchers, you know what impact factors are very important. So I just wanted to put this slide, and I'm very happy you could share my slides to your participants, whoever want. No problem. You can make it, whatever distribution. So all these things are important, and when we have a multinational authorship, obviously that creates a higher impact. So now, earlier, Dr. Santosh mentioned about Global Eye Genetics Consortium. So one of the things that a few years ago we did, and Dr. Natarajan was one of the founding members of Global iGenetics Consortium team. I was the founding president. Currently, president is Dr. Takeshi Iwata, and then you have Dr. Paul Baird from Australia. He's the vice president, and Professor Calvin Tang in Hong Kong. He is the chairman of scientific committee. And clearly the idea was to share genetic information and rapidly isolate within unique populations, establish the system for accurate diagnosis. These are all, again, we wanted to make it a clinically useful issues. Sorry. Develop a cost-effective genetic analysis, develop genotype-phenotype relationship, support relationship, and work with Asian, African, South American, and other countries where not a lot of work has been done. When you look at the current data, where does most data come from? White Caucasian population, right? Clearly, what we want to do is bring to a level everybody else, because that's where the answer of a lot of eye diseases will come. Suppose if somebody finds that, one of the person was recently telling me, we were at SAO meeting in Delhi, and somebody told me that, oh, they know a family of 3 generations, and there is diabetes, severe diabetes, but there's no diabetic retinopathy. So what is going on there? There must be some gene somewhere which is quite playing an important role, turning it off. Think about it. You have seen that too. And how many generations do you? At least 2 generations. So see it's such an important information. Now think about it. If you isolate it, I don't think there's one specific gene. Or you understand a couple of those genes or wherever, and Dr. Kumudi is going to talk about, get into more genetics issues. Imagine how many people around the world will benefit if they made the product out of it, they made a diagnostic about it, if they make biomarkers about it. That will help the whole world. We haven't found that in that part of the world where most of the data currently comes from. So this part of the world will have to participate in order to give the benefit of this knowledge to the rest of the world. I mean, Dr. Santosh is finding that patient population here, in Pune. So there's a gold mine in Pune for not just for India, for the rest of the world, for everybody, right? So there are many examples like that. Let's move on to that. So what are we targeting? We are targeting, oh, before I do that. So one of the goals is that what Dr. Natarajan and I, Dr. Takeshi Iwata, and we organize that, we want to organize and hold regional meetings. That's what we are doing here right now, right? So this is all part of, and then all of you are participating, or most of you are involved in organizing the Global iGenetics Consortium, the first international meeting. My kudos to your team, actually, that you guys have really been working hard, and we are going to make it. We were going to have that honor at National Eye Institute at NIH campus, May of 2020. Guess what happened in May of 2020? The whole world shut down, correct? So now you get the honor. You will be holding the first international meeting of the Global iGenetics Consortium. So I admire you. You're ahead of this weekend meeting. My kudos to you and my salute to you for actually taking up the task. So thank you for doing that. So if you look at it, we want to promote the networking. We want to promote the small collaboration, publications, submission of research grant, and so on. These are very doable things, just at the ground level, so that we can all come up to that level to have the rest of the world benefit. These are some of the pictures who have been compiled for the last 10 years or so. And of course, you'll see Dr. Natarajan is pretty much in, every picture. And if he's not in the picture, Dr. Natarajan is taking the picture, right? The very first one, you see that here, this is when the original agreement was signed between my boss, Dr. Paul Seaving. This is the picture here, number 1, in Tokyo, Japan. And this is me here, and the president of Tokyo Medical Center, director of National Eye Institute, Dr. Paul Seaving, and Dr. Takeshi Iwata, who is the current president. This picture was taken by, credit goes to Dr. Natarajan. Of course, he was the witness of that, right? And Professor Miyake, yes. Oh yeah, of course, yeah, Professor. Yeah, that's so, yes, yeah, of course, yeah, this is Professor Miyake. sitting sitting next to me. Okay. And again, most of these pictures, they're from around all over the world at various places. This was in Delhi, and this was in Vancouver, and so on. And there are, you can imagine that in his database, he has pictures, even more pictures from around the world. So let's move on to the next thing here. So in terms of, we have been working together for many, many years. We have established centers, we have established collaborators. One of the big things that has happened, we brought the whole world, literally whole world, on just one single platform. And this publication, the series Advances in Vision Research, Volume 1, Volume 2, Volume 3, last 6 years, we have produced 3 volumes. And currently our team is working on Volume 4. So the goal is to really bring everybody together in terms of research, single platform, and this is becoming a very prestigious publication. Of course, the proceeds, we don't personally benefit. It all goes to the Global Eye Genetics Consortium. Now, I want to talk about the training quickly. So we are spending a fair amount of, and we were there at the SARC Nations Summit, actually, just the last few days in Delhi. And of course, I wanted to put down this, look, there's a lot of information that's coming from SARC. So we are actually having many early clinicians, early stage researchers come to work with us in the US. They're coming to NIH for training. And there were many people, lady who, Dr. Shalja, you'll meet her she was trained by us actually, and she did just putting together her own research team. She just submitted a new research grant. And I was so pleased to see that we have been part of actually mentoring these people. On the right-hand side, you see that these are the people who are working in our laboratories for 2, 3, 4, 5 years as a PhD researchers. And Indian researchers are about 40% of that. So many of these people are coming back to India, setting up their own labs. Perhaps you could invite one of them to actually set up a genetics lab in Pune. So I'm sure if somebody is local and who is getting training at National Eye Institute wants to come back to, perhaps you can all build a consortium. We'll set up a lab here so that you can have your own genetic analysis and so on. And there are students, and I'm happy to help actually. So I have to give you some information where there are current opportunities. There are research grants available. And people can look at the U01 grant. There's an Indo-US grant collaboration that is very well established. There are over 12 projects that are running. There are training grants available. So this is kind of an important slide that will help you submit and share with your colleagues, establish your own beginning for research as well as training. And our grants are available to citizens of all over the world. You don't have to be an American citizen to apply for these things. Many people in India have applied, and Shankar Netrala has received 2 big programs, Arvind and LV Prasad, Shroff Hospital, these people, AIMS, these people have been running these programs for a number of years. Now it needs to come down to the next level. People need to work with small places or smaller places or medium-sized places like Pune. So that's where the answers. I have a really strong feeling a lot of answers will come when we meet with the unique populations. Okay, so I also want to be very proud of announcing this, new competing grant, which we just released a few days ago. And this program is being given to University of Florida combined with IIT Kanpur. And as you can imagine, this is really not a clinical program. This is a structural landscape of photoreceptor synapses. So imagine that, India is making a contribution in understanding structural biology. Who would have thought of that, right? But so this is something to be really proud of. And, my origin is from India, as you said, Dr. Santosh, in your background. This makes me, and this is a competitive worldwide competition. So we should all be very proud as a world citizen that Indian scientists are making huge contribution. Of course, Dr. Bardman, you were in Florida, and this Florida organization is actually working with them. So such a relationship can be built here as well, right? The next due date would be November 8th. Last week we had the grant submission date, and I know several people in this country, they have submitted the new grant proposals. So you can aim for submitting something. So, 8-10 months to think about it. But I would like to tell you that, it just takes several months of hard work. So if you're going to do this, start early, start 6 months, get in touch with me. I'll give you all the resources. We run workshops, we work with Government of India, and we'll help you in many ways. But you have to start, you have to do your work, right? We are not going to write grant proposals for you. Okay, so at the end, I would like to say that, global science is very interconnected. We are actively involved in not only research, but training. We support essentially everything from basic discovery to translation to clinical research. We seek scientific opportunities. We support US domestic grantees to work with people outside. And anyone from any country in the world can apply for NIH grants. NEI team, we look forward to working with you. So with that, I always like to give this message. This is a public service message, which, I morally, personally feel responsible that it's very important to not only, you are all eye doctors, so I don't have to tell you this, but essentially I like to tell people, hey, watch your eye health is very important, right? And being an Indian originally, I like to tell them, actually at World Sight Day, I gave a worldwide lecture, and I started, my first slide was Sarve Indriyanam Nayanam Pradhanam. Think about it, how important it is, and you practice it every day. So these things are all very important, and even small precautions, small things can make a big difference in life. Anyway, I want to thank you very much. Thank you, Maharashtra Ophthalmological Societies, the local organizers, the rest of the people who are joining through Zoom or video. Thanks to all of you. And I like to say that good eye research anywhere, good eye research in Pune is good eye research everywhere. Thank you so much. Time-wise, we are okay.
Okay. So, yeah, right. So we are open for question and answer session. So those who are on Zoom platform, they can also ask their questions. Dr. Kaumudi, do you want to ask questions about research grants? And yeah, actually it is a thought-provoking lecture by Dr. Gyan Prakash. In fact, very interesting. Nobody knows much about research grants and what can be done. It's a huge ocean, and actually we have not tapped even a small dust of that ocean. So a drop of ocean also we have not tapped. So probably in next few years.
There's one thing, my father used to quote that Lord Hanuman didn't know his own capabilities. Right?
Yes. Yes.
So you don't know your capabilities. Somebody has to be there to remind you of your capabilities. Think about IIT Kanpur just received a huge multi-year grant. Think about it. Think about it. Isn't that amazing? To do that. They're not doing any basic, any small type research. They're going to be submitting, and they fought, and they actually won a worldwide competition.
That's really great. We are happy to know that. Only challenge as a practicing ophthalmologist or a practicing clinician is that to dedicate time and finances, managing the finances. That is a challenge, but I'm sure with the research grants and other things, we will be able to do a lot of research from this part of the world, especially India. We have great minds and we are very intelligent people. So I hope that
everything I agree.
The next Nobel laureate will be from India. Especially in the,
we may be looking at
genetics. Actually, I was looking at Dr. Natarajan because that's his dream. I know that is his dream.
And we want to be there with you. He promised that he will take me to Stockholm with him.
So, does anybody have any questions from the audience in the Zoom? I think we can take questions later on.
Okay.
So we will.
So the last part of your question, your last part of the question, I think that it really ends very well because that's where Dr. Natarajan and Dr. Kamala come into picture. So they'll tell you more of the ground level view. So thank you again. Thanks very much.
Thanks for excellent talk again, Dr. Jan Prakash. And our next speaker is Dr. Kaumudi Godbole. She has done her MBBS from BJ Medical College, Pune, and MD from KEM Hospital, Pune. She has finished her MD in pediatrics in 1994. After that, she was a lecturer, and she has done her training in genetics from University of Toronto. She's a fellow in clinical genetics from University of Toronto, and currently she's a practicing clinical geneticist at Dinanath Mangeshkar Hospital, Jahangir Hospital, and KEM Hospital. So I request Dr. Kambudi to give a talk on gene therapy. Where are we today? Sorry. She's going to talk on eye opener on genetics for ophthalmologists. So her talk is more for us. Dr. Kambudi, please.
Right. So good evening, and thank you for involving a clinician and a non-ophthalmologist in this auspicious, I would say, meeting. I say auspicious because it's really, really unusual in clinical setups to have clinicians, administrators, researchers all on the same platform. And it's very important because we all feel when you're in your clinic or in your office or your lab that you're doing the best job. And you're not really understanding what is somebody else's problem when you want them to collaborate with you or to cooperate with you. And I think such kind of platforms give you an opportunity to discuss, and that would be just a beginning. And I'm not sure who all attending this meeting, but I assume everybody attending is ophthalmologist primarily. Yeah. So some of the slides you might find that are really, really very basic. And do you want me to?
It's not working. No, it's not working.
We can go in the slideshow and do it.
Okay, why is this happening?
Yeah, so this
Take it.
Slide share.
Would it be enough? But I'll have to take this down.
Okay, I'll take that.
You'll have to take this down.
Yeah, I'll take it down.
Take this down. Now we have to
Slideshow.
go to slideshow. Yeah, this one.
Yeah, no, okay.
Slideshow. And then, okay. So it is.
And 1 minute. Yes. It's all right. Okay, take this one. Take this one. I'll take it. I'll take it. It's, see over here, you can see it's pretty fine.
Yeah, but I don't know why that button
doesn't
went.
work
It was
there
there in
also.
the button here.
This is one more. This is screenshot. Just reduce. One. That's it. Leave it. Leave it.
Okay.
It's visible.
Okay, so I don't know what exactly. I mean, there might be some parts of the slide that you might not be able to see, but then you can imagine. And so now it's just going down. I'll have to possibly click every slide.
No, no, 1 minute.
Yeah, I think now it came, so you can. Good. Okay, so as I was saying that some of the slides might be very, very basic, but I think that would be a good revision. So why do we need genetic diagnosis? One is because genetic etiology is important in isolated as well as syndromic eye disorders. Many a times people feel that genetics is all funny looking kids and all kinds of abnormal things. You could have completely otherwise normal looking people who might have genetic disorders, and it is important to find out what the problem is. exact diagnosis helps in accurate prognosis and management. It's like fever, you have fever, it could be typhoid, it could be dengue, it could be anything. So in your field, it could be retinal detachment, it could be myopia, it could be strabismus, but the prognosis and the associated features might be very, very different. And that might depend upon the genetic etiology underlying. And it offers possibility of identifying at-risk family members and prevention of transmission of the disorder to future generations. I think this is very important. We all are very excited about gene therapy and all the advances that are happening in the field of gene therapy and treating genetic disorders. But we cannot forget the bottom line of how many of us are actually going to use it in clinics. How many of us are actually going to prescribe it in a clinic? And that's why prevention is very, very important aspect of genetic disorders, at least maybe in this century, or at least next 10 to 20 years, I would say. And it is basically every patient's right to know the information. And that's why it's important that we keep pace with the genetic advances. So now I do just enter, or
Next slide.
here, right side, okay. So it's really sometimes very scary when you look into that small eye. We used to be scared as students and that's why we didn't go to ophthalmology. We didn't take ophthalmology, such a small organ, but such a complicated organ and one of the most important sensory organs. And all the genes, the developmental regulation, it's a very, very complex interaction because there are multiple embryonic tissues that are involved in making the eye. There are multiple genes that interact with each other, and they also have inputs from the environmental insults that might be happening during that period. There are genes involved in extraocular tissues also. So there is a diverse structural and functional defects of multiple structural components, which are governed by multiple genes that interact with environmental factors, which makes it rather difficult and interesting maybe to study eye genetics. And I'm not going to go into details of these busy slides, but this is basically to tell you that there are lots of different embryonic tissues, neuroectoderm, neural crest cells, surface ectoderm, mesoderm, all of them contribute to the making of the eye. And if anything goes wrong with any of these tissues, any of the tissue development, there are going to be birth defects. Similarly, it's a long drawn process. Starting at day 25 and continuing up to 9 months and even later, the formation of the structure, so the hardware and the software of eye is a long journey. And anything that happens around this time, so it could be some teratogenic drugs that the mother consumes, or there is high blood pressure and the mother gets a convulsion, or there is neonatal sepsis, or there is some neonatal hyperbilirubinemia, let's say. All these things are going to hamper the normal development and maturation of the eye tissues that are very important for us to function normally. There are lots of genes that are involved in eye development, and some of the genes might be more familiar, like PAC6 or 6-3, and some genes we don't really very commonly talk about because there are some genes which cause too many problems because they are involved in too many structures that make the eye as well as some other tissues of the body. And there are common genetic phenotypes. So just let me make it clear that I'm not really going to talk about a particular genetic disorder or a particular phenotype. This is just about an approach how to suspect a genetic disorder and what we can do about it. So 6-3, as you can see, can lead to cyclopia and arrhynia, holoprosencephaly, really severe manifestations. And maybe practicing ophthalmologists don't really see many of these cases, but we see it many often as geneticists, especially in prenatal genetics, because we see pregnant ladies who are carrying fetuses who have these kind of problems. And we do fetal autopsies regularly for such fetuses and then identify various other problems. And also we see neonates who are born with such problems. And then there are certain relatively less severe defects, which the ophthalmologist actually see in their clinics, because the person has to first survive and then they can go to ophthalmologist and then medicine and any other specialties that are necessary. So as I said, PAX2 is important, and most of the genes that are involved in lens, various proteins that make the lens, or proteins that are, or transcription factors, they are going to create similar phenotypes. So if you just take cataracts, there'll be lots and lots of genes that cause cataract, which otherwise is such a, I guess people say bread and butter of ophthalmologists, but it's a headache to geneticists because there are too many genes involved in cataracts. And you think that it is an isolated condition, but the same gene could be implemented in syndromic cataracts. One might think that it's an autosomal dominant condition. That means only one copy going bad is good enough to cause the disease. But in some other patient, you see 2 copies affected and the person has got a different disease, the gene being the same. So it's interesting and difficult at the same time to get into genetics. And there is a nuclear genome and a mitochondrial genome, just to remind you. So nuclear genome, as everybody knows, and I think this was the end of our genetics knowledge when we studied in 80s and 90s and so on. So we all knew that there are 46 chromosomes and 22 pairs of autosomes and a pair of sex chromosomes, etc. And whatever was contained in those chromosomes is it makes the nuclear genome. So all our genes that we typically talk about, Mendelian inheritance, those are all located on these chromosomes. And then there is a mitochondrial genome, which is maternally inherited. We need to remember that only the heads of the sperms penetrate the ovum to fertilize the ovum. So every one of you sitting here have your mother's mitochondria. The father's mitochondria doesn't enter because they were left behind in the tail that kind of led the sperm to the ovum. So mitochondrial disorders are transmitted from mother to all her children, but an affected male cannot transmit it further because his mitochondria are not going to make a baby. And there is a variable concentration of normal and abnormal mitochondria in every cell. Just to remind you, there are lots of mitochondria in every cell, and we have so many cells. So many mitochondria, some of them might be normal, some might be abnormal. So the severity of the disease, onset of the disease, and the tissue which is going to be affected. All of you, I think, remember that mitochondrial disorders, they can affect all high energy requiring organs. It could be heart, it could be kidney, it could be brain, it could be eyes, and anything can come up first at any age. And you really have to be very, very lucky to spot that disorder and also try and identify it. So very quickly going, previous one. Okay, so Mendelian inheritance patterns. Now, again, this might be very basic to some of you because we have some knowledge of inheritance patterns, Mendelian inheritance patterns. Now, in all these figures, the dark is unaffected and the light part is affected. Okay, and here you see typically in consanguineous marriages, there are, in autosomal recessive inheritance, there are consanguineous marriages or marriages within the community. And you see that parents are not affected. So here the key is the circles are females and squares are males. If it is fully shaded, it is an affected person. If it is half shaded, it is a carrier person. And the double line between that man and a woman is the marriage line or mating line. And the line which connects to people from above is a sibship. So there are sibs, and then there is a couple that you can see in this figure. And then there are 4 children. One is affected, 2 are carriers, and 1 is normal, as you can see in the progeny. And so whenever it is an autosomal recessive inheritance, generally the parents are not affected. From outside, they look normal. And we all call ourselves normal just because we don't get into genetics. Because at cellular level, at molecular level, none of us is really normal. All of us harbor some or the other minor variations in our DNA. Some of it might be trouble causing. Some of it might be just a silent change. And we need to remember that in genetics, we try not to use this term of normal and abnormal when we are talking to patients, because we understand that none of us are normal. And Then comes the autosomal dominant inheritance, where the disease can be transmitted directly from an affected person to his progeny, 50-50 chance, or it could be a new dominant mutation. So nobody has the disease, like say Marfan syndrome or achondroplasia. So where parents are normal and you see the child having an autosomal dominant disorder because it's a new disorder that happened in the child. We all know that none of the children are Xerox copies of their parents. Their genes and their chromosomes combine and an entirely new baby forms. So every baby has new things to get. And sometimes there are mistakes that nature cannot or forgets to correct, let's say simply. And those are new sporadic mutations. And here you see that the father is affected and two of his children are affected. And you see both girl and boys to be affected. So we are talking about autosomes, which have nothing to do with the sex chromosome. So both boys and girls can get affected in autosomal disorders. While if we come to X-linked conditions, where female is a carrier. Now, as you all know, both, female has two X chromosomes and male have one X and one Y. So a female can carry on even if she has a mutation in one of her X's because the other X is working well. So she will not manifest generally. Of course, there are examples like ocular albinism, where you can look at the female, the fundus, and then think about albinism because you see signs there, although she is not going to manifest with the any other features. And in X-linked condition, you see that female is an obligate carrier. So that small dot is a indication telling you that she is a carrier. And she has passed on her X chromosome that has the mutation to that affected son, and the father has passed on the Y chromosome. I guess everybody understands this, that for a boy, the X chromosome has to come from the mother and Y chromosome has to come from the father. And so this boy is affected, his sister is a carrier. And there is another sister who's not a carrier because she got the good X from her mother and the good X from the father. And the other boy, who also is normal, got the normal X of the mother. So all possible combinations are there, and we need to find it out when we are dealing with X-linked disorders. Now, this was X-linked recessive. And because males are hemizygous, that means they have only one X chromosome. So there cannot be two X chromosomes, one copy affected, one not affected. So the point to remember is in X-linked recessive inheritance, females are asymptomatic. While in case of X-linked dominant inheritance, Alport syndrome, if some of you remember, or incontinentia pigmentae, such disorders are X-linked dominant, where females can also get affected because their only one affected copy can make them sick or make them have the disease, and they can pass it on to males. So in every generation, you are seeing affected persons. And but the gene is located on X chromosome, so it's an X-linked dominant condition. And then we come to non-Mendelian inheritance patterns. We just talked about mitochondrial disorders. And just to show here in the pedigree that the female is affected, all three children were affected because they all got a big load of abnormal mitochondria. The male stopped sending those mitochondria ahead. So his children were normal, but the females continued because their mitochondria will go ahead. So just by drawing pedigree, we learn a lot. We can get an idea what we want to do, what sort of inheritance we want to look. And thanks to Human Genome Project, we think we know about 20,000 genes that we have and what they're doing and which genes we can concentrate. I mean, suppose you want to do research on autosomal recessive retinal detachments or any of your phenotypes, then you know which genes are going to get transmitted. So you can ask your clinician colleagues to give families or samples which have this particular inheritance patterns in their families, so that the sample will be kind of a homogeneous sample and the chances of finding a causative gene or causative mutations, founder mutations will be very high. Because if you have, for example, thalassemia, in India, 90% of thalassemia is caused by 8 common mutations. There can be 200 mutations or even more in thalassemia. But most of the labs will test for these only 8 common mutations, and they will kind of screen 90% of the population. That's possibly not as easy in cases of eyes, but I'm sure if not 90%, we will have some mutations which are seen in 10%, 20% of the population being so huge. So the panels could work even in those conditions. And then there is something called co-dominant inheritance, or blood groups, where both the A and B alleles express themselves. Nobody is dominant, nobody is recessive. So if it's an AB blood group, both A and B express, and they don't overlap each other. There is no intermediate between A and B. Both are there. And we don't really have many diseases of co-dominant inheritance. This is just an inheritance pattern. And those of you, if you are interested in this talk and it's you're awake, here's a question. So Mr. LA is diagnosed with some type of retinal dystrophy. One brother and sister are similarly affected, and 2 of his brothers are normal. His parents are also normal. Now there's a possible diagnosis. I write possible because there are no documents, and which is often the case in Indian situation. Many a times people burn all their documents. If the previous baby dies, they don't want to keep any memories, and they just throw off all the important investigations, and you are also at loss. Anyway, so this person doesn't have any documents, but he's suspected to have some retinal problem. And this is the person, his maternal cousin's son, maternal aunt's son. And he has a normal brother and sister. And this maternal aunt and her husband are sister and brother of his father and mother respectively, making it complicated, right? So what is the likely inheritance? So the person where there is an arrow is this LA. He has 2 affected brother, I mean a brother and a sister. 2 brothers are not affected. And his parents, the brothers and sisters of the parents, have married each other. So nobody is consanguineous here. It is just that they are brothers and sisters, that's it. So nobody is affected. I mean, no parent is affected. There is no consanguinity. Girls and boys both are affected. So what could this be? So this is an autosomal recessive pedigree where the parents are normal. And very often we see this, and it doesn't have to be that somebody in the family has to have the disease, because it's a very common misconception that the moment you utter the word genetic, it means that, but nobody in our family has a genetic disorder or has a diagnosis. Possibly because the parents are carriers. Now, maybe you are tricked here because I didn't show the carrier status of the parents. I just tried to draw what was told in the pedigree. So what are the mechanisms of genetic disorders? When we come to nuclear genome, there are chromosomes, and there could be chromosomal defects. There could be single gene disorders. So there could be aneuploidies, which is abnormal number of chromosomes, which is Down syndrome, trisomy 13, trisomy 18, and so on, or Turner syndrome. Structural variations, which could be either microscopic, which you can see under the microscope, so karyotype, which all of us understand, or they could be sub-microscopic, for which you need even further zooming in. And the current investigation of chromosomal microarray does wonders in finding out these very, very tiny deletions and duplications. Then there are single gene disorders. And as we just discussed, one disorder, many genes, and one gene that can cause many different disorders. So all sorts of combinations are possible. Variety of mutations, missense mutation, nonsense mutation. Those of you who are used to read research papers related to genetics, missense mutation is just a change in that DNA sequence which still makes some protein. Nonsense is kind of stopping the journey there. No more protein is formed. Out of frame, now if you remember codons and all that, 3 nucleotides make 1 codon. And so if the pattern of 3 is interrupted, no proper amino acid will be formed, and then the protein production will not happen. There could be whole exon that can be deleted. Just to give an example, DMD, Duchenne muscular dystrophy, there are 79 exons, and there can be deletion or duplication of the whole exon. So exon is the coding region of the gene. and there is triple repeat expansion. So in a condition like Fragile X syndrome. So I think I won't go into the details of this, but there are many different mechanisms which, if you're interested in genetics, most welcome to read. Epigenetic changes, UPD, that is uniparental disomy. Normally, we assume that our genes are equally contributed by mother and father, half-half. And that's what is supposed to be. But if there are situations where both the genes have come from only the father or only the mother, some of them are imprinted in the sense they will function only if they have come from father. And if they've come from the mother, that gene will be silent. So assume that both the genes have come from the mother. Then the paternal gene will not be there, and there will be, it's almost like not having that gene function. So conditions like Prader-Willi syndrome, where such situations can happen, or many other imprinting syndromes. Mitochondrial genome, and then multifactorial conditions, which I think most of the ophthalm research is going on. All the gene environment interactions, diabetic retinopathy, and so many disorders where environment, especially smoking and alcohol, and some genetic predisposition. So these are all multifactorial disorders. So just to show the chromosomal disorders, then there could be single gene defects, and neural tube defect is one such multifactorial disorder. Epigenetics is above the genetics. Now, somebody just told me we're trying to figure out what genetics is, and now you're talking about epigenetics. But I think we need to understand that, because that's what we face every day in our lives. Our genes are not hidden somewhere. They are constantly interacting with the environment, and their expression changes. So what's this? This is a flute. All of you have seen that. And unless somebody plays that flute, there is no music. So if our DNA sequence is there, it is intact, there are no mutations. Unless the gene expresses itself, it is not going to produce proteins. So there are cancers because of overexpression of certain genes, or there are cancers because of suppression of the controlling genes like oncogenes. If they get suppressed, there will be growth that is just not stoppable. So similarly, there are mechanisms by which the DNA will get activated. There are factors. All the transcription factors that you talk about in genetics, they help in expression of the gene. And only then the gene product will be present. And that's why it's important to keep a track of epigenetics also. Genetic tests are all special because you have not heard about them during your medical training. And that's really very important, because the moment we start interacting with other specialties, everybody is kind of very disturbed, even just listening to the names, forget about using them. They're costly, they're time consuming, and they're available only at certain labs in the country, which I think is not a hurdle anymore. The labs are very happy to come to your doorstep to collect samples. So that is not anymore a problem. Require explanation and informed consent. Now, typically as clinicians, we are used to just scribble something, hemogram, urine routine, cholesterol, and the patient goes, and patient himself or herself can read that report. This is the normal range and your report is so and so. Nobody, a lab doesn't tell anybody that you don't have hemoglobin. But such things can happen in genetic test. A genetic test can come back saying that we didn't find any genetic change. Or it can say that we found a change, but we don't know what it means, because no other patient has been reported to have that problem. So it's a variant of uncertain significance. It's not benign, it is just uncertain. So you have to wait till there are functional studies or some other patients who also again get that. And as the previous speaker was mentioning, unless we have our own data, unless there is contribution from this part of the world, so many of our real variants are going to be classified as variants of uncertain significance. Now, there are more than 10 labs in India which are doing a huge work in whole genome sequencing, whole exome sequencing. Previously, I guess previously is about 7 to 8 years back, I used to send samples to labs in US. And practically every time the report would come back as a variant of uncertain significance, because the Caucasian data set didn't have that variant. And then the functional studies prove that, okay, if you change this amino acid, the stability of the protein will not be there, or the gene will not function properly. And eventually we started getting more Indian, Asian samples, and our database is also increasing now. And most importantly, there has to be pre and post test counseling, because it is so difficult even for the doctors. How will the patients understand what you've done after spending about 20,000 rupees that they gave you? So it is very important, and it's your duty to tell them what is possible, what the result is, what it means for them, and only then one should order genetic testing. And there are types of genetic testing. Some are just screening tests, which I don't think we have many specifically for ophthalmology, unless we consider metabolic disorders like galactosemia. We have a screen. So if somebody is screen positive, then we go and investigating further. And there are some diagnostic tests, and those diagnose the diseases which are currently present, or they can diagnose the diseases in future. So if somebody has a mutation present today, but the disease of onset is going to be late onset, then maybe if there are any preventive measures, they can take care and try and help. Of course, there are few disorders. I think in ophthalmology, I can't think of many examples. Like in breast cancer, you could do certain surgical treatment or self-breast examination and try to find out if breast cancer is going to actually happen, and get it treated early. Huntington's disease, there is no prevention, even if you knew it. But these are all predictions, and but they diagnose the condition. Okay, so these are various tests that I have all included in one slide, and then there is one dotted line. So the JIVAS, which is at the left-hand bottom side, that's mainly a research tool. And whichever diabetes research paper you take, there will be a JIVAS conducted on 35,000 or 50,000 samples, because they are going to group people affected and people not affected, and trying to figure out which genes are predisposing to a particular condition. They do not diagnose, they don't find out causative genes. They just say this group of genes which have small effects are all important or could be important. So we don't use JIVAS in the clinics. That's a research tool. Everything on the left side of this red line is talking about chromosomes. So karyotyping. FISH is a test where you need to know the locus, exactly the address of the gene or that particular part of the chromosome. 22q11.2 microdeletion, the DiGeorge syndrome, or Williams syndrome. You need to know exactly which location you want to look at. And on the right side is gene sequencing, which used to be the Sanger sequencing, which used to be the only tool we had, or when the human genome sequence was being discovered. And now we have exome sequencing, where we can do all 20,000 genes in one go. And technically that's possible even in one day. Although in clinics, we get the reports in 4 weeks or 5 weeks, because we are not researchers, we are clinicians. So they have to be doubly checked, and very, very stringent protocols for clinical information. So just taking congenital cataract as an example, and I will possibly give more examples related to pediatric population, because I'm primarily a pediatrician. So, and because lens development continues throughout the intrauterine life, also in later life, cataracts can develop at any time actually in somebody's life. 70% are isolated, 30% with associated features, and 15% of them have extraocular features. So something wrong with the hand or kidney or heart, some other tissue involved. And therefore, multidisciplinary management is definitely important to reduce the comorbidities, and early surgery and close follow-up helps them. I think you know all these things much, much better than me. And bilateral cataracts are often associated with positive genetic mutation, and autosomal dominant mutation is most common. And all types of cataracts have been noted. And crystalline mutations are the most common, because they're really very important part of the lens. And routine genetic testing is essential with next generation sequencing. That means not Sanger sequencing, by doing the whole exome or whole genome, because there are so many genes. So we don't know. We can't just look at a cataract and say, oh, this is cataract because of this gene. This is just cataract. The gene could be any.
Servicing केली ना चांगली?
Sorry. Okay. Right. So prenatal ultrasound can detect congenital cataract in the second trimester of pregnancy. So what you can see on the right side top, that's an ultrasound, and the arrows are showing congenital cataracts. So we have seen cataracts at 20 weeks, and obviously parents want to know what would be the vision. And I don't know whether right and wrong, but there are some parents who don't want to continue the pregnancy if there are cataracts in the fetus, because even if we say that cataracts are operable and the vision could be near normal, nobody can guarantee that the associated features are absent. Because in fetus, you can only see structural defects. You can't see functional defects. You can see 2 ears, but you don't know whether the ears hear. Similarly, even if you think that the cataracts can be operated, how will the brain function? Because brain and eye is so close. Yes, and any disabilities, and it is the parent's choice, at least in India. The rules change everywhere. Yeah, most of the conditions, even today, hearing loss, sorry. Well, ideally previously it used to be 20 weeks. Now it's up to 24 weeks, but it has to be a real solid reason, not just for one CTEV or something. So, right, but thankfully we are happy with 24 weeks, because we have, at least in all the mega cities in India, very regularly ultrasound follow-ups are done. Most of the anomalies are diagnosed by 20 weeks, and we are getting most of our results before 24 weeks. So we used to crib a lot when it was 20 weeks, but with 24 weeks, we are quite happy now. Now I'm showing this slide just to scare you, just to tell you that there are so many more genes identified for cataract plus extraocular features. So there are many ophthalmologists who feel that, well, we don't really see the rest of the patient. We are concerned with the eyes. But if you take a look at the whole patient, you might find out there are many other problems that the child has or that the person has. And sometimes you have to look for the problem. The patient is not going to tell you that I have only one kidney, because it's possible that you're living whole your life with one kidney without even knowing it, unless you have had an abdominal ultrasound for some reason. Or sometimes there is a tiny extra finger which the parents have removed when that person was a child. So he also doesn't remember that he had an extra finger, and it is such a minor thing that nobody discusses, but it could become a syndrome if with that small, with extra fingers, an eye and something more going on. Okay. So I just want you to remember this one syndrome, nothing very specific, but we have seen a handful of cases where the diagnosis was given as congenital rubella embryopathy because there were cataracts, microcephaly, and this actually is a genetic condition, very severe and horrible genetic condition where the child just doesn't develop well. There are lots of problems: microcephaly, microcornea, cataracts, developmental disability. And practically everything that you see in birth defects can happen in this syndrome for the eye. And MRI brain shows agenesis or partial agenesis of corpus callosum, pachymicrogyria, hypomyelination, cerebral atrophy. And there are 4 genes. When I kind of found my first patient with collaboration with UK scientists, there was only 1 gene, and possibly that was the one case that we had. Of course, unfortunately, we didn't report that case, but now there are 4 genes, and both autosomal dominant and recessive cases are found. And please do not miss them as congenital rubella infection or perinatal hypoxia, because that is the favorite diagnosis of Indian clinicians in general. Whenever you see hypointensities or hyperintensities in the MRI, a telltale history, it's very easy to say maybe it was birth asphyxia or perinatal asphyxia, and that's why the child has all sorts of problems, but it's not always like that. So these are all the associations: face, brain, kidney, skin, musculoskeletal, and one actually needs to look at all these things if you find one eye abnormality. So this is a girl. These examples are all from internet. I had to find some quick pictures all clubbed together. So she has those tubers in the genes. Then this, I think all ophthalmologists would know, this is Treacher-Collins syndrome with down-slanting palpebral fissures and limbal dermoid and retrognathia that you can see on the profile picture. And there are many genes that cause Treacher-Collins syndrome. Now this is charge association. Now charge association has now become a syndrome. Why? Because a syndrome is supposed to have a known etiology. When we say some features are common together, but we don't know the reason, we say it's an association. But when we know the reason behind that association, it becomes a syndrome. So now we know that there is CHD7 gene that is causing this association. So we call it Chart syndrome now. And that's Marfan syndrome with lens dislocation and retinal detachment, and there is fibrillin gene. And then this is a condition with brittle cornea. There are many different genes which actually lead to brittle cornea. And there are repeated perforations, and there could be skeletal abnormalities associated, blue sclera and skin scars. Then an Alport syndrome where you see lenticonus and the flecky retinopathy. I don't know what has suddenly happened to the font here. Sorry? Yeah, yes. And then this is telangiectasia, as you can see, ocular telangiectasia, cerebellar atrophy. So this is ataxia telangiectasia. And just some more cases, the oil drop cataract that's supposed to be seen in galactosemia. We see children much before. We see newborns which are there and in the nursery or in 15 days. So thankfully, because of newborn screening, we prevent all these complications. So nowadays, I think hopefully we don't see cataracts and other things in galactosemia, because we hope to prevent it by altering the diet. Niemann-Pick disease, where you see cherry red spot and the brownish discoloration. Albinism with diffuse chorioretinal atrophy, cystinosis, and so many of disorders. As I said, I'm not really going to discuss these disorders, just giving examples that there are so many things that actually have a genetic basis and we need to look for it. And these are movement disorders that can be present in many late onset conditions, many retinopathies and many other conditions which can actually have nystagmus. It can have affection of the saccades that you see, or it could have some sort of oscillation of the eyeballs and so on. But on the left side, there are a lot of illnesses or lots of disorders that have been listed. And if one is interested in adult onset or late onset genetic disorders, we need to look not only at abnormalities, but abnormal functioning of the eye structures also. And this is an interesting registry. I'm not sure whether in India we have, because I'm not a routine visitor of ophthalmic genetics, but this is one registry, and people can directly contribute, and so they are trying to generate data of what sort of distribution they have of retinal dystrophies. And I don't know what will happen in India, how this pie chart will differ, because I'm sure our genetic background is different, and our retinal dystrophy pattern also might be different. So what is the American Academy of Ophthalmology clinical statement? That because there are causative mutations in almost 75% of the patients, we need to do genetic studies. A negative result does not rule out genetic etiology. It is because we might not yet have discovered that gene, or the data interpretation might change once that mutation is found in many other patients. And as we saw in the earlier slide, it is important for diagnosis, for prognosis, for helping the family. And if there is gene therapy, to treat with the gene therapy also. There are age-related macular, I think everything is changing, all the font, and anyway, as long as you can read, I'm happy. So there are lots of gene environment interactions in all late onset disorders. And macular degeneration, now we know that there are therapies which can include antibodies to certain growth factors. And because eye is an easily accessible organ, it can be directly injected into the eye rather than giving it through complex systems and then worrying about whether it is reaching the eye and whether it will cause side effects somewhere else. So eye is possibly one of the ideal targets for gene therapy. Yeah, looks like. And it is sporadic. I mean, in some, yeah. So diabetic retinopathy, all I wanted to say is almost all genes that are involved in causing diabetes have been blamed also in retinopathy. So really, from my knowledge, I don't think there are only a few selected genes that if you have them, then you are more likely to develop retinopathy. There are because these genes are going to interact with the environment. It will depend on what is the diabetes control and at what stage diabetes happened, and so many different things. So currently it is more of research rather than helping therapy, but I'm sure unless you do research, there cannot be therapy. So it's again egg and hen kind of a story, and we all need to be patient to get that first result. I remember I think I have completed MBBS in when I don't even remember, maybe 90 or 91. That time we used to say that DMD, and we thought that, and we used to hear from people that people are researching on DMD, Duchenne muscular dystrophy. And here now we are in 2022 with actual drugs for certain exon deletions, duplications of DMD. So the first always takes more time and more efforts. But I think once there is an opening, many more drugs are all in the pipeline. And I think in next 10 years are going to be very, very exciting for genetic medicine as such. Mitochondrial disorders, we just touched. So gene therapy, there's a talk on that. So I'm not really going to say anything more than that because I haven't really done any gene therapy myself. I don't have experience in that. But as I was saying, retina is really, really an important place where gene therapy can do wonders because of its immune state, special immune status, no lymphatics, direct blood connections, and so on. And currently available gene therapies for certain retinal dystrophies and lawn and so on. So we'll listen to that in the next talk. And I'm just going to finish my talk with a few more slides. One to tell you about prenatal diagnosis, because I think you don't see that in your practice. So many prenatal ultrasounds are going to detect all these various types of birth defects in case of eyes. So from 12 weeks of gestation, the person who's doing fetal sonography is supposed to look at the fetal eyes. And then there are special views mentioned. And magnetic MRI also helps because it gives a better resolution. And whenever there are these kind of problems, generally fetal medicine and genetics are involved because we need to sort out what is this leading to. So you can see prenatal pictures and then pictures after birth. You can see that there is one eye normal and the other eye not formed. So there's a cryptophthalmos, and then there is a proboscis there. And in the lower picture, you can see bilateral cataracts. You can see the ultrasound and also a 3D image of ultrasound in that picture. This is the prenatal MRI. If you're not aware, we do fetal MRIs from 20 weeks of pregnancy. So 20 to 24 weeks is a good window to look at the fetuses, get their MRIs done for especially neurological features and even eye. So how and what to test? So genetic testing is a must when we are thinking of doing a genetic diagnosis, and there has to be counseling before and after the test. The labs are at the doorstep, as I said. We need appropriate sample, filling up the forms and taking an informed consent. And informed consent is very, very essential in genetic testing, which we clinicians are really bad at. We just say, you believe in us and I believe in you. That doesn't work in research, and that doesn't work in genetic testing either. So results take time, and they're not very easy to interpret. So you need to take help of your colleagues. And so when you suspect a genetic disorder, go for genetic consultations, do investigations. If there is a diagnosis, treat the patient if there is a treatment, or assess the at-risk relatives. And if there is no diagnosis, wait for more research, then you might be able to help. And now I'm going to finish with this case, which is for you all to take a look at, which I can't read here, but as I remember, this case is of a girl. About 20. Now she, when she came to clinic 2 months ago, she was about 25 years. She was operated at about some 6 or 8 months of life, and then she has generally been asymptomatic. She also has retinal detachment in the same eye, but the other eye is perfectly normal. She has another young sister. Her parents are non-consanguineous and they are asymptomatic. And she was the first person to have retinoblastoma. And so here are the questions. What is the chance that she carries a germline mutation in the RB gene? All of us know that many retinoblastomas are sporadic. They are not inherited. So when we say germline, it has to be present throughout the life, and it can go to your next generation through the gamete. So that is a germline mutation. So are there any, now this is all ophthalmology. So what is the chance that she has germline mutation? So what information do you need? For that, there is no family history. So typically retinoblastomas have a germline mutation if there are many affected relatives, or if it is a bilateral tumor. But here it was a unilateral tumor. However, it was multifocal. The notes that she carried those 25 years back, it said that there were 3 discrete nodules in that same eye, and I don't remember the exact techniques that they did, but surgery couldn't be done, and she was some other treatment was given to her. And what test you will order? So the first thing is MLPA, which is a type of test which detects deletions and duplications in the RB1 gene. But in this patient, no abnormality was detected. Then we did the gene sequencing and we found a mutation. And as you can see, it is a heterozygous mutation. Heterozygous meaning only in 1 copy. If it is in 2 copies, it is homozygous. So RB1 gene in a way is a dominantly inherited condition. So it is also a single mutation that gave rise to retinoblastoma in her. So this was the report. So there was a mutation in the exon 17 of the gene, and the variant is given there. So a variant is like a typo, let's say. T is typed instead of C, and then the whole amino acid the DNA sequence changes, and the amino acid produced also changes. And then it is written that this particular gene is associated with retinoblastoma. It is an autosomal dominant condition, and the classification of the variant is pathogenic. That means it is surely going to cause disease. And this is the interpretation, that it results in a stop codon. So it will stop protein production. So a truncated production will be there that can cause disease. So the next question, what is her risk of passing on the gene to the offspring? We said it's an autosomal dominant condition, so 50% risk. And will you offer prenatal diagnosis? And the problem here is this gene doesn't have 100% prediction. So if you carry the RB1 gene, there is kind of a 90% possibility in your lifetime, and because this is a blastoma in early life, to get a tumor. But if the fetus at 3 months is found to carry the mutation, what if the fetus actually is in that 10% who's never going to develop retinoblastoma? Will you still terminate the pregnancy? And thankfully, this decision is supposed to be taken by the parents, because as doctors, we cannot ignore that 10%. So you do the best of your counseling and let the parents take their decision. Most of them generally do not want to go ahead if there is a mutation, because there is a 90% possibility that the fetus will actually eventually develop disease. Is her younger sister at risk of having affected offspring? So ideally, you need to test her since in all her, I mean, retinoblastoma is not going to happen when she is an adult. So most probably she is not a carrier, but if you want to confirm, you can do the testing and complete the story. So that's it. Thank you. I think I have gone over time, but thank you for a patient listening. Thanks. We can take questions.
Yes. Thanks, Dr. Kamudi, for an excellent talk on genetics from ophthalmologist perspective. And now I request Vardhaman to introduce our favorite, Dr. Natarajan sir.
Thank you so much. I think just before I introduce Dr. Natarajan sir, I would like to ask a question to, I think, everybody who is on the Zoom and everybody who is here, which was actually showcased in the last presentation as well. So which was the first gene successfully targeted in ophthalmic gene therapy in the history? Can anybody tell? Which was the first gene which was successfully targeted, and there is a US FDA approved first time therapy for that gene. So which is that gene? Natarajan sir knows, of course. All right, anybody who is there on Zoom, anybody can like probably try to predict if they know. Okay, so this was the RPE 65. So this was the one which was first gene targeted, and the gene therapy was approved by US FDA. All right, so that brings us to the next topic, which is extremely relevant to us as ophthalmologists, that where we are today in terms of gene therapy. And I think in this regard, we are lucky to have Dr. Professor S. Natarajan sir with us to give this talk. Of course, sir doesn't require any introduction, not only in India, but all over the globe now, in ophthalmic community especially. Everybody knows that he's a vitreoretina surgeon par excellence since 1984. He's a Padma Shri awardee, which is one of the highest civilian awards given by the President of India, and also especially the state award by the government of Jammu and Kashmir for his service when there were a lot of pellet injuries that he had treated. He has done humongous, more than 60,000 plus vitreoretina surgeries, has given more than 2,500 invited guest talks all over the globe. He has authored more than 4 books and has peer-reviewed publications, which are more than 100. He is the charter member of the Retina Hall of Fame, which is very prestigious honor. He is currently the president of the Teleophthalmic Society of India and also the Asia Pacific Ophthalmic Trauma Society. He is the worldwide secretary general of this global eye genetics consortium, under which we are conducting today's CME and also the international meeting next weekend. He is a board of trustee and treasurer of the International Council of Ophthalmology, and I believe this is the first time an Indian has become a board of trustee for ICO, which is very prestigious honor. He is a board member of the International Society of Ocular Trauma. He is a Guinness Book of World Record holder for screening maximum number of diabetic retinopathy patients. He is also a trustee of the Kamala Sundaram Foundation, under which we are conducting this CME today, and which is the NGO serving economically weaker section and doing free eye checkup camps, and have done so far more than 4,900 camps and have screened more than 2 lakh slum houses. His dream is to prevent blindness due to diabetic retinopathy especially, and make India blind free in the next 5 years. And for that, he has initiated a registered Section 8 company. He's also a trustee member of Roti Bank, and which serves about more than 10,000 free meals every day, especially in Mumbai. So over to you, Dr. Natarajan sir. Welcome to POS and MOS. Thank you.
At the outset, I'd like to thank Dr. Santosh Bide, President, Maharashtra Ophthalmic Society, Dr. Jay Gelkar for Poona Ophthalmic Society, Vardhaman, and also Anaga, I think, for Maharashtra Ophthalmic to take this topic. I think actually I'm interested in gene therapy for long. One is in 1984, my father started the genetic eye clinic in Government Eye Hospital in Madras, which is the oldest eye hospital of India and second in the world. And also Dr. Kumar Manikwel, who's known to me and NEI, and Dr. John Prakash was part of the team. And his professor, Dr. Ratna Savarodi, was a veterinary geneticist. He helped in starting that. So from that time, I'm interested, and we made the first paper along with Dr. Kumar Manikwel and my father, and presented in Asia Pacific Academy of Ophthalmology in 1985, and Dr. Arthur Lim. At that time, I met him, and I'm a friend of him from that time. That's why I'm in the board of APSCRS also. And so the genetics I'm interested because when I did my postgraduate and when I wanted to join ophthalmology, the retina fellowship, half the people, or not half, including my father, our entire faculty in government eye hospital said, half the book of Parson, you can't do anything. And why do you want to do retina fellowship? And I think surgically, we are doing so many things for diabetic retinopathy, traction detachments, and then another half or maybe quarter of the book was genetic eye diseases, and it's inherited retinal degeneration. So I'm happy. 2017, I started the inherited retinal degeneration clinic at Ajjo, and all this inspiration, even though it came from my father and grandfather, thanks to John, who really encouraged. Otherwise, I was looked at what can you do as a researcher in front of Shankar Netrali or L. Prasad or Ames, and I think maybe same or maybe better than them. And all the rankings, Ayatollah is included. So what is gene therapy? Introduction of exogenous DNA into a host tissue to produce a therapeutic protein, peptide, or RNA. So history of gene therapy in 1940s, nucleic acids were identified. 1960s, methods using bacteriophages developed. 1970s, recombinant DNA technology. And 1990, first gene therapy clinical trial approved for children with severe combined immunodeficiency. And why eye is suitable for gene therapy? I think excellent talk by Dr. Poona, wonderful, because more than 200 genes are responsible only for inherited retinal degeneration. That's why I'm looking at next 10 years, I can do only inherited retinal degeneration surgery and probably earn good money for the hospital as well as good care for the patient. And easy access to the site requiring the gene via injection. You need a vitreoretinal surgeon to inject the gene vector into the subretinal space and that too near the macula. My friend, she is Dr. Fanny, originally born in Pondicherry, now a professor in Ghent, and she invited me. I'm going in November 23rd to the Belgium Ophthalmic Society. She's one of the pioneers working on robotic surgery for doing this gene therapy for RP65. I mean, it's only 800,000 US dollars, almost 8 crores for one eye. And partial sheet from immune system assures reduced risk of destruction of vector carrying the therapy. And that's why it's unique. There's a retina blood barrier, like the blood-brain barrier. And as Madam said, the exogenous, during the embryo development, you have the neuroectoderm and surface ectoderm meet, and then the eyeball comes. And that's why I still remember my anatomy lecture, even though it was in 1976, we had a professor, M.M. Cooper. He was an excellent anatomy professor who taught us very well, particularly the embryology. So the common inherited retinal dystrophies are RP, which all of us know, Leber's congenital amaurosis, which we just heard, and cone dystrophies. And many times anybody diagnoses anything and they don't know what is the diagnosis because nobody looks into it. And there's a famous saying, I forgot the author, I should Google it. Whatever the mind does not know, eyes do not see. And I remember my father teaching me, ophthalmology is the only field you should not do anything blindly, no blind procedure. And macular dystrophies, you have Stargardt dystrophy, Whitley-form dystrophy, familial drusen, pattern dystrophy. And that's my dream, and I'm happy that in the Retina Hall of Fame, I'm along with Stargardt's and Warren Helmholtz, who found the ophthalmology. Thanks to Dr. Badrinath for the wonderful pure metasmic. And gene therapy goals, identification of new target genes, development of viral vectors that permit delivery of large gene payloads, initiate strategies to treat conditions that have polygenic basis. The problem is even monogenic we are not able to treat, and now they're thinking of polygenic also, that means multiple genes. To develop programs that not only rescue patients with the existing condition, but therapies that delay, prevent, or permanently cure eye disease. In a lot of disease we say we don't have cure. Thanks to my friend Subur Huang is also having a foundation. There's a function, I think on November 19th, they give an award for this sort of contribution. And how retinal gene therapy works. The vertebral neural retina contains many different types of cells like the retinal ganglion cells, rod and cone disc photoreceptor. When I did my postgraduate, I mugged up the 10 layers of retina. And we never knew that we will do an OCT, and we can do an OCT angio now. And I remember in 99, when I wanted the first OCT, we got it in 2000. And We brought the first photodynamic therapy to India. Dr. Badrinath asked me, what are you going to do with this machine? You're wasting money, he told me. I said, no. I said, something will come. And you can imagine every general practitioner may want a OCT now. And without OCT, they can't see the macula. So I think that things differ. And I think that's why I learned this first in Maharashtra, first in India, first in Mumbai. It's a game, but I think it's worthwhile to be in that game. So I introduced a lot of firsts to India, and I'm happy about that. Same thing for gene therapy. I want to be the first in India to get the gene therapy. So how do we start working? So I'm training my staff. There are optometrists are coming and going. Ophthalmologists are coming and going. They forget to make a detailed history. And every day I have to break my head on teaching them pedigree chart. Dr. Fielding, HMAC from National Institute, thanks to again Dr. John Prakash, brought him to Bombay. We had a meeting and he taught us that the pedigree chart can be downloaded from the internet. But most of the things are paid. But I think why not? Nothing is free. There's no free lunch. And that's what I have to learn from America. Something is there if somebody is giving you free because you have to pay for it. Comprehensive ophthalmic evaluation, which is needed for every eye patient. Genetic counseling, which people don't know what it is. And when I tell a patient, go to genetic counseling, they don't want to do because right now, because of COVID, the previous genetic counselor left and we are doing it in online. Patients say, I'll do it later. Consent and collection of blood sample. The biggest hurdle I'm facing, every patient tells me, what are you going to do with the blood? You're going to do research. And I'm having this problem from 90 because we did a public meeting and collected a lot of blood samples for Kumar Manikwell. And comprehensive includes proper visual acuity recording. So all of us, Dr. Santosh said, he's a practitioner, he doesn't have time for research. No, he can do for every patient. Only maintaining a record is important. When examining a patient is important and documentation is important. And that's what is finally somebody can analyze it. You don't have to sit and analyze, Santosh. You have to employ optometrists, pay only one more optometrist from your profit and tell them to do only research. That's what I'm doing. So I'm fed up with the surgeons who are coming to learn retina surgery because they want to make money. I know this is going in YouTube and internet everywhere, but I think it's fact. I think people want to learn some technology only to make money. No, I think my father told me, you can make money, but for making only money, my friend is here. So Shah is there, he's a builder. Thanks for coming. And he is a great friend of ophthalmologist because I think his father is a trustee of Pune hospital, right? What hospital? Poona hospital. So thanks for coming. And so we have to do the central peripheral vision defects, confrontation, ocular motility, arm's-length grid, ocular health assessment with slit lamp biopsy. And people keep on sending photographs, OCT, and ask for opinion. So now I have a new idea. So yesterday only I wrote, somebody from US is sending. I told them, why not you get a slit lamp biomicroscopic examination and ask that surgeon only to give the opinion. How can I sit in India and give an opinion, I wrote. Because I'm actually angry. What is the point? Just because they know me, and then the patient has undergone retinal surgery by a great retina surgeon. Now something is there, red in the eye. I said 3 things are there in the eye: pain, vision, and redness. This covers the entire ophthalmology, including genetics. Contrast sensitivity, glare testing, VEP, ERG, EOG, all these patients actually they think I'm making money. But anyway, I tell them it's for your benefit. If you want to do it, do it. Otherwise, it's up to you. And I keep it in the record. And color blindness, recent research have found out that AAV can successfully restore color vision to treat color vision blindness in adult monkeys. You know, this is what is scientific marketing by the Americans and Europeans. And it's not available for the humans, but one day it can happen. So never say never again was Charles Kippens, who's the father of modern retinal detachment and guru of Dr. Badrinath. So he was guru's guru. So I'm also a member of the Kippens International Alumni. And I think he told me this. I'm happy he has visited my clinic. An entire retinal stalwarts have visited. So juvenile retinal cases, and we heard Dr. Kumudini is talking about various genes producing the degenerative disease. AAV5 vector containing a wild type human RSI or a cDNA driven by a mouse opsin promoter showed long-term retinal function and structural recovery and improvement in the retinal structural reliability. Again, thanks to Dr. John Prakash, who encouraged me. I attend RO every year, and I think I was also in the RO mentor group. And RO is only for researchers. But one good thing is anybody can attend. Whoever is interested in research, whether a physicist, chemist, geneticist, pediatrician, ophthalmologist, cardiologist, anybody can be there. An X-linked journal retinosis, we have a foveal cyst. Like this, they will say foveal reflex dull, diagnosis not known, close the chapter. And this happens in government medical college, happens in private practice. It happens by retinal surgeon also. So an RP, and I think I'm not going to spend more time. We all know it happens in all the three gene mutation, the single gene, the X-linked, and also autosomal recessive. It's a peculiar. And in that, 200 genes are there for RP alone, and only RP65 we have a treatment. And that's costing, unfortunately, $800,000. That means almost 8 crore for one eye. And this, you can see the defective gene, RDH11. And I think you have to go to RO to see what all happens. And inherited eye disease is characterized by irreversible photoreceptor loss, and the activation of resident microglia is secondary to the disease. Stem cell has been tried. We have a friend in Kapil Bharti. I wanted to go and see him, and he's doing a lot. He also worked with a company called iStem in Bangalore, and a company which they have a branch in San Francisco, and they got an award from the government of India recently for an. I think they are working on some stem cell therapy, but I don't know when it will come. And then this prevent disease by inserting a gene into patient's cell instead of using drugs or surgery, replacing a mutated gene that causes disease, delivering the healthy copy of the gene. It's exactly like 3D printing, copy paste, and also CRISPR is there. You cut the gene and delete, add. So inactivating or knocking out a mutated gene that is functioning improperly, introducing a new gene into the body to help fight a disease. And here you see the natural virus carrying the therapeutic gene, infect and deliver therapeutic gene to target cells. This is the great part. Whatever is negative, virus, everybody knows viral fever, there's COVID, and I think even a common influenza is a virus thing, but we are using this for treating. So you can convert that, and I think that's where I'm happy Dr. Abdul Kalam and Dr. Manmohan Singh said religion and science are convergent and not divergent. So I think it is also it's a spiritual level, and you have to work on the cellular level. And the most common viral vectors for cell, it's an intravitreal injection. It didn't work. And subretinal injection targeting photoreceptor and RP. And that's where my role as a vitreoretinal surgeon comes. Also access from the suprachoroidal space. Both are only with the vitreoretinal surgeon and not, fortunately, with the cataract surgeon. And the advantages, a minimal immune response, capability to mediate long-term transgene expression in a variety of retinal cell types, AAV serotype initially used, and 8, the new variant, have increased efficiency of gene transfer process. And generally, subretinal AAV is more efficient than the intravitreal route because the subretinal route may be subject to greater degree of immune privilege compared to the intravitreal route. And virus as vector, active clinical gene replacement trials are targeting Stargardt's disease, Usher syndrome, retinitis pigmentosa, XLRS, that's X-linked retinoschisis. Thanks to Dr. Paul Sieving, who's working on it. He was working in, he just told me, I met him last month or first week of November, and he told me that he's transferred the research from National Eye Institute to the UC Davis, and he's expecting to start the clinical trial. And I told him, think about India as the first, because he knows me. Thanks again to John. He brought him when he was the director, and he's going to bring the current director who's going to address our Maharashtra ophthalmic. Santosh, you should be happy because director of NEI is addressing us on 19th. And also we have achromatopsia, Leber congenital amaurosis 2. And these trials use either adeno-associated virus or lentivirus as viral vectors. And this is what Dr. Vardhaman mentioned, who knows about the first gene therapy. It's approved last year, February, but it's 800,000. So according to me, like this IIT Kanpur or IIT or any of the basic science institute, whoever can go through this YouTube video one day should do some research in India. Definitely we have to collaborate. And I wanted to say, diabetes has multiple genes. Mohan Vishwanathan's Diabetic Research Center, his brother Dr. Vishwanathan Center, Shankarnetralaya, Arvind are working. And last week I'm part of the Lancet publication by Rajiv Raman and Shobha Shivprasad from Moorfields, where we have contributed something on the genetics, because 20 centers did this study called Ornate India, And I'm trying to collect a few crores for doing that through my new foundation called Sundaram Natarajan Blind Free India Foundation. It's a Section 8 company. As you see here, they have actually connected a syringe and putting it in the subrectal space. It's not so easy. And you have to do vitrectomy surgery, you have to expert, you have to avoid complications because every procedure in vitrectomy surgery has complication. And if you read the side effects of this gene therapy, there are 10 of them had inflammation, so which is published in Nature. You have to read a lot, and that's why I think Vardhaman's wife asked me, how do you become younger? I think you read a lot. You read because every day you learn. And so you can see the various layers of retina here, where the virus enters cells, healthy gene released to cells, and cells return to normal function. I think even to make this diagram, they have spent, I think, an amount of time, crores or in dollars. I think amazing. I think you have to salute America instead of only criticizing on McDonald's and other things. Same McDonald's gave the inspiration to Dr. Venkatasamy to do $10 a cataract in India. And you can see this is a list of conditions which you go to, again, thanks to clinicalresearch.gov, which is maintained by federal government of US. And I think there's a lot of things to learn. And Spark Therapeutics is doing this RP65 Luxturna and marketed by Novartis. Can believe I asked them to start this in India? So that's following the corridor. So this is Dr. Robert McLaren's video. Dr. Mike. So this is how the vitreoretinal surgery is done and subretinal injection is injected.
Recording in progress.
Their parents found an experimental treatment, and 2 years ago, the kids enrolled together in a clinical trial. It was with Spark Therapeutics, a young biotech company formed out of research from the Children's Hospital of Philadelphia.
The idea is that we are taking the correct functional copy of the gene and adding it back into the cells that have the functional or missing copy of that gene.
It's called gene therapy, and it's relatively uncharted territory in medicine. The technology uses a modified virus to deliver a healthy copy of the gene to make up for one that causes disease. Scientists have been working on it for decades, but only now are the first gene therapies approaching the market. Sparks is among the most advanced. The healthy genes are delivered through eye surgery. Once they're in place, they're designed to do the work of a normally functioning gene, meaning patients only undergo treatment once.
So step.
Had trouble seeing in low light. Now, with our vision perfect, it's big differences.
The first patient actually was brought to RO in the meeting itself. Jean Bennett was the person who did it. So thank you. And I think same thanks to Dr. John Prakash and Takeshi Iwata, who's the president of current Global Eye Genetic Consortium. They visited me many times. We started a lab in Thane, thanks to my late Dr. Vasudev Ginde, who ran the diagnosearch. He's a close friend of Dr. Patwardhan, retina surgeon. What's his full first name? Ardhyadhar Patwardhan. So he's very close friend. They have studied together, and unfortunately he died at 55. I think as a medical doctor he didn't take care. But anyhow, he gave me a space for lab, and Takeshi came and said, this lab is better than my lab. But actually no, the building was great and the equipment was very, very little we had. Even Sudha Iyengar, whom I'm doing a genetic research with on diabetic retinopathy, where we are working with the slums. 4 years we are struggling, writing the grant, and no influence works. So no, and we have to really be deserving it. That means compete with the rest of the world. And I don't know how many crores we have. I think maybe 11 crore is what we have approached for the research. What will be the result? We don't know. It will take another 8 years. We are trying to find a biomarker so that we can classify which patient we have to concentrate to follow up and which patient may not come for follow-up. We don't know still. 25% of diabetics will get diabetic retinopathy. And today is World Diabetes Day. And if you read the research of Banting and his student, both of them got the Banting and Best. And the Best was a student. And I believe 2 residents were there, and one of them took a lottery who will work with Banting. And then they got the Nobel Prize too. And actually, Swami Vivekananda died of diabetes because there was no insulin. He died in 1902, and the insulin was in 1927. So that's why I wanted to introduce this subject, which is close to my heart, not only to propagate vitreoretinal surgery, but to make patient, public aware. And that's why I am inviting everybody to come for this conference, both online, offline in Mumbai. It's on 19th and 20th. You can visit gegc.org. And we have experts talking from US, Japan, India, China, Australia, Brazil, South Africa, Turkey, and maybe some other countries too. I think I recently have included a talk from Mexico where I met a clinical geneticist and trained in again US in the last month when I went for live surgery. So we are also planning to apply to the government of India with BIRAC, Department of Biotechnology, starting clinical registry for inherited retinal degeneration. Dr. She asked about whether there's a registry. We have a free software from Dr. Takeshi. He's also delaying giving it to India. He suggested that we will start an Indian genetic registry, but unfortunately, everybody is worried about identity, which he promised that there will be a masking and you can't, the patient identity will not be given. And I am still working closely with him. I hope to get that platform developed. He has got 35 universities sending blood samples to Tokyo. And thanks again to Dr. John, as Tokyo Institute of Medical Sciences signed with NEI. 2 years later in Yokohama, they signed with Aditya Jyot. And that's why they keep coming. We had our own fellow. She's a researcher, MSc. Now I have about 10 of them. Somebody is a BTech, somebody is a BSc. So we have a lab in National Facility for Biotechnology, which was inaugurated by our Prime Minister in Khalsa College, and which we run. The lab is small, but I'm happy. Hope to, as Santosh said, we have to work for it, whether I get the prize or not. And I think to win or not to win, but to participate seriously so that the patients will benefit. So first line is to create clinical registry for inherited retinal degeneration, because in 5 years, if the treatment starts, you cannot just treat any inherited unless you identify the gene. And we have to have funds. In America, I think, where was this? Where was Donald Gass working? Which the university? Where Dr. Anita Agarwal also was there. No, she moved. Vanderbilt. Vanderbilt chairman is Paul Paul Stegman, I forget the name. He was the president of RO also. He made that all the genetic testing in that institute, Vanderbilt Institute, free for the patients. And because they have data, and the same thing India should do. Somebody has to sponsor and get all the genetic database so that patient can be given free, and also give it to the patient what has been tested. We are, and she mentioned about age-related macular degeneration. Takeshi and working on age-related macular degeneration on ARMS2, the gene which you mentioned, one of the 20 genes in ARMD. And we also did, and he worked with the company to block that ARMS2 gene. But we had a PhD in genetics, Dr. Rajkumar. Anyway, everybody wants to have more income, so they left me. They've gone to another company. But I will keep, like I'll keep on getting up, like a kid, falling down, getting up and climb. So in summary, vision loss from inherited retinal disease is debilitating, and genotyping is the key to developing possible gene-based therapies. So as Dr. Kumudini said, that phenotype, what is that phenotype? That means all the clinical examination, particular test with the ERG, EOG, color vision, autofluorescence, and then you diagnose, clinical diagnosis, that is phenotype. Then you do genotyping, that means genetic testing. She mentioned all types of genetic testing, and where you have the genotype. So then you call, after knowing the genotype, it may not be a cone rod dystrophy, it may be some other dystrophy. So we can label it after gene therapy, and we call it re-diagnosing after gene therapy, after genetic testing. And patients don't want to understand that, because now the private company MedGenome, and I forgot the other names, they are doing testing, but they charge, and patients don't want to pay. So unfortunately, we live in an era where everything they think they can buy. They can buy the mother, they can buy the father, they can buy the child, and that's a problem. And I think we have to change our thinking, because even if you want a child, I tell them, healthy child, you have to wait 10 months. And the main target for ocular gene therapy involves the retinal pigment epithelium or photoreceptors, and most common method to deliver viral vector is with us, with the intravitreal surgeon, subretinal injection delivery, or suprachoroidal space. So these are the references for, I leave it here. Thank you very much. And I think we're already late. Maybe people who are in Zoom are finished their dinner, and people in the US maybe in the morning finish their breakfast. Thank you. Thank you very much.
Thank you, sir. I think that gave us a nice understanding of what is clinically happening around the world. So I think from Maharashtra Ophthalmological Society, I will take this opportunity to thank all our faculty. Dr. John Prakash, sir, he has come all the way from Washington. Thank you for being here as part of this special CME on eye and genetics, and telling us about all the research opportunities. Dr. Kumudini Godbole, ma'am, thank you so much for giving us some basics. It is like revisiting the medical school again for us, and I think we all learned a lot from your talk. And thank you so much, Dr. Nadi sir, for not only your amazing talk and giving us a great overview about what is happening in gene therapy, but always inspiring all of us with your sheer presence. So thank you to everybody, all the delegates who have joined here physically, everybody who has joined us on Zoom and also on YouTube and Facebook platforms. And I would like to also thank our past president, Dr. Ladi sir, and past president of POS, Dr. Gitanjali ma'am, also to be here amongst all POS and MOS members. Thank you again. Thank you, everybody. And we see you next weekend for the first ever coming weekend on the first ever international meeting on eye and genetics by the Global Eye Genetics Consortium. This is happening for the first time in the world. This was supposed to happen in the US, as we have heard. We are very honored that we have opportunity, and MOS is part of this group, which is under which aegis we are planning this. So we look forward to see all our MOS members, all our POS members for this meeting as well. Thank you. Yeah. Yes. So I think especially we have Colonel Shankar from Army who is there, and Colonel Sandeep Kumar who is there, and Mr. Subhav Shah, who is a very close friend of Dr. Nadi sir. I have also had opportunity to meet him in the past, and a great foodie. That is what I know. So, good night, everyone. Yes, thank you so much.